Many of the most effective treatments for cancer and autoimmune diseases rely on a single, vital tool, the monoclonal antibody. This is a microscopic, Y-shaped protein engineered to hunt down specific disease targets. But moving these proteins from a lab bench to a pharmacy is expensive. Citing the high costs of research and development, the price tag to launch one successful biologic drug typically sits between $2 and $4 billion. Despite those costs, the approval rate is low. Only 5 to 10% of antibody candidates are ever approved for the market. In this industry, 9 out of 10 drugs fail. The drug discovery pipeline operates like a high-stakes gamble, where the vast majority of promising candidates never reach a patient. A major cause of that failure rate is a lack of developability. This refers to the ability of a drug candidate to survive the transition from a controlled laboratory setting to large-scale industrial manufacturing. During mass production, fragile antibiotic structures face intense physical stress. They are mixed with high shear force, constantly agitated, and exposed to significant temperature shifts. If the antibody is unstable, the damaged proteins begin to stick together. This is aggregation, and it turns the liquid into a dense, high-viscosity goo that is unsafe for injection. These macroscopic failures aren't random. They are driven by microscopic flaws hidden deep within the drug's primary amino acid sequence. Because these physical flaws often go undetected until the manufacturing phase, companies can spend millions on a candidate that is physically incapable of becoming a product. To identify these risks earlier, researchers are using biopharmaceutical informatics. This field uses software to stress test drugs in silico before a single drop of liquid is ever manufactured. It starts with structure prediction. By using machine learning tools like AlphaFold and databases of known proteins, researchers can predict a drug's three-dimensional shape simply by analyzing its linear sequence of amino acids. The challenge lies in the variable regions, the tips of the Y-shape. These are the parts that actually bind to a disease, and they require highly detailed modeling to understand their stability. These digital clones allow scientists to find structural flaws in a virtual environment, identifying failures before they become expensive real-world problems. With the 3D model established, the software runs a scan for physicochemical liabilities, the microscopic motifs that trigger protein degradation. One major risk is the presence of hydrophobic patches. These areas repel water and act like magnets, grabbing onto other antibodies and causing the clumping that ruins the drug batch. The scan also flags chemical instability. If specific residues like tryptophan or methionine are exposed on the protein's surface rather than tucked inside, they are prone to oxidation when they hit light or air. Researchers also use isoelectric point calculations to predict if the drug solution will become too thick to flow through a standard needle. By identifying these red zones virtually, scientists can predict the macroscopic behavior of a drug in a vial using its microscopic sequence data. Once the flaws are identified, the drug enters lead optimization. This is the engineering phase where scientists surgically edit the antibody's code to remove those liabilities. This flowchart shows the complexity of the task. On the left, researchers must remove specific sequence liabilities, while at the top, they have to maintain overall developability and stability. The right side of the chart highlights the final constraint. The drug must be humanized to avoid an immune response and maintain its activity, meaning it still has to bind effectively to the target. It is a systematic balancing act. By patching the antibody's weak points, researchers turn a fragile discovery into a robust therapeutic product. This engineering results in an antibody that maintains its therapeutic function while meeting manufacturing standards. It is no longer just a laboratory hit. It is a stable candidate that stays in solution and can be reliably produced at scale. For decades, connecting microscopic protein structures to macroscopic pharmaceutical behavior was a major unsolved problem. Biopharmaceutical informatics is finally bridging that gap. This diagram illustrates the shift. On the left, the traditional discovery process is a scattered approach, relying on trial and error. The right side shows how informatics consolidates efforts, focusing resources on a candidate that meets the criteria for large-scale production. As researchers develop complex multi-specific drugs, the trial and error methods of the past become unsustainable. Transitioning to computed developability creates a more efficient path for translating lab discoveries into reliable medicines.